Hello everyone, today's lecture is going to be about photosynthesis. In our last lecture, we discussed about respiration and all of the topics related to respiration, and today we will discuss about photosynthesis. So photosynthesis is one of the most important chapters, but in the MUR era there has been less focus on photosynthesis chapter. You can say it was really high yield in Cambridge era, but in MUR era there are less MCQs from this chapter, but still if the respiration has been focused for 2024 and 2025, that means they might shift towards photosynthesis chapter 2. So this is a chapter that you can't afford to skip. Okay, so respiration and photosynthesis are always studied together, so you can't afford to skip this chapter. So I will be discussing about these important topics that are about light dependent, light independent reactions, about chloroplast, what are photosystems, what are C3 and C4 plants, and what are the factors affecting photosynthesis. So if you want to know what are the high yield concept lists, then you can take a screenshot of this page and you will know what you have to study and what you have to skip. If you have taken the screenshot, let's start our chapter. So first of all, this is the introduction of photosynthesis. So what is photosynthesis? So photosynthesis is the process in which energy poor inorganic oxidized compounds like carbon dioxide and water, they are reduced to energy rich carbohydrates like glucose, and they use light energy that is absorbed and is converted into chemical energy by chlorophyll and some other photosynthetic pigments. So the process of photosynthesis is actually in front of you. As you can see, the carbon dioxide here, carbon dioxide is being converted into glucose. So this carbon dioxide is reduced to glucose, so reduction is happening here. And if you see towards the water molecule is being converted into oxygens, so oxidation is happening here. So oxidation and reduction, both of them are happening at the same time. That's why photosynthesis is actually a redox reaction. So photosynthesis actually happen in two stages. The first is the light dependent reaction and the other is light independent reaction. So light dependent reaction means that it depends on light and it occurs in the thalakite membrane, and the light independent reaction is also called as Calvin cycle, or you can say dark cycle, right? And it occurs in the stroma of the chloroplast. So we will be discussing these two stages in detail, but before we go into these stages, you have to know what is the link between photosynthesis and respiration. So as you know that what happens in respiration, so in respiration you make ATP and NADPH, and in photosynthesis you make glucose. So photosynthesis and respiration, both of these uses the product of each other. Like photosynthesis uses the products of respiration and respiration uses the products of photosynthesis. But there is a difference here. The difference is that photosynthesis occurs only during the daytime, while respiration goes on during day and during night. So what actually happens here? So as you know that during day only photosynthesis happens and respiration happens all the time, and during dark, when there is night, leaves and other actively metabolizing cells respire and utilize oxygen and release carbon dioxide, but photosynthesis does not occur. And during dawn and dusk, when light during at dawn and dusk, when light intensity is actually very low, and during that time you can say the rate of photosynthesis and rate of respiration where for a very short interval of time become equal to each other, and thus you can say that the oxygen released from photosynthesis is just used and just is required for cellular respiration, and the carbon dioxide released by the respiration will be just equal to the quantity that is required by photosynthesis. So you can say like the oxygen released from photosynthesis will be used by respiration, and the carbon dioxide released by respiration would be used for photosynthesis, and both of them become equal to each other. So photorespiration and photosynthesis and respiration would become equal to each other. This is actually called as compensation point. What is it called? It is called as compensation point, just like this. So before we move on to our next topic, you have to understand what is chloroplast, what is photosystem, what are chlorophyll, because without understanding these topics, you won't be able to understand the whole process of photosynthesis. So the first process that you need to know is about what is happening in the chloroplast. So you have already discussed about chloroplast in detail in chapter number 4, that was cell structure and function. So in chloroplast there are actually two membranes. There is outer membrane and there is an inner membrane, and the space between these two membranes is intermembranous space. And then you have thylakoid membranes and you have granas. You know that a coin-like structure that is present in the stroma of the chloroplast is called thylakoid. Stroma is actually just like the cytoplasm of cell. Similarly, stroma is the place where all the thylakoids are scattered in the chloroplast. So stroma is just like cytoplasm of the cell. So thylakoids are coin-like structures that are present in stroma, and when you stack thylakoids in a pile, and then the whole stack of thylakoids will be called as granum, and the space inside the thylakoid is called as thylakoid lumen. If you make thylakoid, this single coin is called thylakoid, and when you keep stacking thylakoids on each other, you will get a bundle of thylakoids, and this bundle of thylakoid is called as grana, right? And if you open up a thylakoid just like that, in that thylakoid, this is the lumen of thylakoid, and this is the membrane of thylakoid, and this is the outside is the stroma. So before we move on to our next topic, let's understand the whole photosynthesis process in the diagram in front of you. As you can see, this is a green leaf, and the leaf there are plant cell, and inside the plant cells there is chloroplast. The chloroplast is open in front of you. There is an outer membrane, this is inner membrane, and this is the space between both of them. And as you can see, the stroma, the granum, the thylakoids, all of them are in front of you. So there are two types of processes that are happening here. The first is the light reaction. So this is the light dependent reaction. So in light dependent reaction, light and water molecules are used and oxygen is released. The products of light reactions are ATP and NADPH, and these products are used in the dark reaction that is also called as Calvin cycle, and the Calvin cycle, as you can see, is happening in the stroma. So light dependent reaction is happening on the thylakoid, while dark reaction is happening in the stroma. So as you can see, the products of dark reaction is a sugar, and the sugar combined together to form glucose. So that is the ultimate product that is formed from photosynthesis, and the other products are NADP positive and ADP that will be used again by the light reaction. Light reaction will produce ATP and NADPH again, and then this cycle will continue in front of you. So these are the two processes that we will discuss in detail in today's lecture. I am just giving you the general idea about how it will happen, where it will happen, and how it will go. So if you have understood the general idea, let's move on to our next topic, that is the photosystem, and the system where all of it will happen. So photosystem actually contains two structures. The first structure is antenna complex, and the other structure is the reaction center. So to understand these topics is really important, because if you don't know what is a photosystem is, so photosystem is actually, like you can say, when so many photosynthetic pigments, the pigments that happens in photosynthesis, they combine together and organize together into a big cluster, and that big cluster, that big system is called as photosystem. So it is a collection of pigments in chloroplast, right? So photosystems are meant for efficient absorption and utilization of light or solar energy that is located on the thylakoid membrane. As you can see, we have already discussed the thylakoids. This is a thylakoid and this is the thylakoid membrane, and the thylakoid membrane contains the photosystem, and this is the lumen. Inside there is thylakoid lumen and outside there is the stroma of the thylakoid. So there are two parts of photosystem. The first part is the antenna complex, the other part is reaction center, right? We just discussed that. So photosystem contains antenna complex, just like the antenna of your TVs and radios, and the other part is reaction center. So both of these parts are really important and they define what a photosystem is actually. So first of all, the first thing is you have to understand what is antenna complex. So antenna complex actually contains pigments, for example like chlorophyll and carotenoids, and what they do is that they absorb light energy. As you can see here, the light energy actually comes and they fall on the green pigments, and the pigments start a chain and transfer the energy, and the energy will move from one pigment to another pigment in the form of electrons, and these electrons will eventually goes toward the reaction center, and this is the reaction center in front of you. So what will the reaction center will do? So the central part, as you can see, is the reaction center, and it contains very important chlorophyll A and primary electron acceptors, and they actually help to produce and in the generation of chemiosmosis. What is chemiosmosis? We will discuss it in detail. It is a process in which energy is utilized and glucose molecules are formed. Okay, so this is the place where all of the reactions will happen in your thylakoid. So light energy will fall on thylakoid membrane, and antenna complex will convert and move that energy in the form of electrons toward the reaction center, and in the reaction center, as you can see, there is chlorophyll A and primary electron acceptors, and both of these will help to make energy ATP, and further ATP will be used to make glucose molecules. So if you have gotten the general idea, now we will start our whole process, because I am just giving you the general idea until now. We haven't started the whole process yet. Okay, so after you are done with this photosystem, you have to understand what is chlorophyll. So you have understood what is a chloroplast, what is thylakoids, and now we will jump on to our most smallest part, that is chlorophyll. So chlorophyll is a really small green pigment that is found in the cells of the plant. And where is it present? It is present in the thylakoid membrane. As you can see here, this is the stacks of thylakoid, and the stack is actually called granum. And in the thylakoid, okay, so in the thylakoid contains so many chlorophylls that are in front of you. As you can see, there are so many thylakoids that are embedded in the thylakoid membrane. And if we take out one single chlorophyll, and this is it how a chlorophyll look like. So chlorophyll contains two types of parts. The first is the porphyrin head and the other is the tail. So chlorophyll are really important photosynthetic pigments of plants. First of all, you have to know that they are insoluble in water. They are really hydrophobic, and they are only soluble in organic solvents like carbon tetrachloride, alcohol, benzene, etc. As you know the basic chemistry. So they are only soluble in organic solvents. They are not soluble in polar solvents like water. The next thing is that it contains many different types like chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D. There are so many types, but we will only discuss about two types, that is chlorophyll A and chlorophyll B, because they are the major types. So if you can see here, what does the structure of chlorophyll actually looks like? So the chlorophyll molecule has actually two parts. The first is the head. It is the porphyrin head. It is also called as hydrophilic head because it is hydrophilic in nature. It is water loving. And then you have hydrocarbon tail, that is hydrophobic in nature. So you can say head is hydrophilic because it is water loving, and tail is hydrophobic because it hates water, right? Philic means loving and phobic means hating. So hydrophobic, water hater, hydrophilic means water loving. So the head is actually made up of magnesium, nitrogen, and it is also called as porphyrin ring. So first of all, we talk about the head. So as you can see here, the head is actually flat, it is square, and it is the part where light will be absorbed. So it is outside the membrane, it is not embedded. The tail is embedded inside that membrane of thylakoid, but head is outside the thylakoid membrane. And as you can see, there is a magnesium ion that is in the center of the porphyrin ring, while the head is exposed on the thylakoid membrane. Similarly, there is a heme group. You know that what is a hemoglobin, right? The hemoglobin is just like the porphyrin ring. So hemoglobin also have the same structure, but there is a difference here. In the hemoglobin, instead of magnesium, as you can see here, there is iron molecules, iron plus 2 molecule instead of magnesium in hemoglobin. So hemoglobin contains iron atom instead of magnesium. So this is the only difference between chlorophyll and hemoglobin. So you can say that hemoglobin also contains porphyrin ring, it also contains nitrogens, but the difference is that hemoglobin contains iron instead of magnesium, and there are 4 nitrogen atoms that are around the magnesium atom. So that is the structure of chlorophyll molecule. You don't have to memorize every single carbon atom, you just have to know that there is a magnesium atom in the center and there are 4 nitrogen around it. And the other thing that you have to memorize it, what are the difference of chlorophyll A and chlorophyll B. So if there is a methyl group, if there is CH3 at the functional group, then it is chlorophyll A, and if it is CHO, or you can say if it is a aldehyde group, then it is chlorophyll B. So this is the difference between chlorophyll A and chlorophyll B. We will discuss it in detail in our table, but you have to know that chlorophyll A and chlorophyll have different structural differences. Okay, so let's move on to our tail section. So there are 2 types of tails actually. First is the hydrocarbon tail that you just discussed. There is another tail that is in front of you here. This is actually called as methyl ester chain, or you can say it is a acidic chain. So it is methyl ester chain. So methyl ester chain is actually small chain, it is acidic in nature, and it is not so important. The important chain is actually the hydrocarbon chain. So hydrocarbon chain actually contains 20 carbon atoms. So the formula is C20H39. So the hydrocarbon tail is actually embedded inside the thylakoid membrane. That is why it's very important. So tail is embedded inside and the head is outside the thylakoid membrane, and head is the portion where light is actually absorbed. So now you understand how does a chlorophyll look like, where it is present and how it is present. So now we will move on to our first topic, that is about light dependent reaction. So first of all, you have to understand where does the light dependent reaction actually happens. So light dependent reaction actually happens in the thylakoid membrane. So there are actually 2 types of light dependent reactions. First of all, there is non-cyclic and there is cyclic light dependent reaction. We will discuss first the non-cyclic photophosphorylation, or you can say non-cyclic reaction. Then we will move towards our cyclic reaction. So light dependent reaction actually have 2 types of photosystems. We just discussed what a photosystem is, but there are actually 2 types of photosystem. The one is photosystem 2 and the other is photosystem 1. So in the first place, the first photosystem that receives the lights is actually photosystem 2. They are actually named according to their discovery. For example, you are thinking like, why the reaction does not start with photosystem 1 but photosystem 2, right? And this is the photosystem 1. So why are we not saying this photosystem 1 and this photosystem 2? Well, the reason is because P700, or you can say photosystem 1 was discovered later and photosystem 2 was discovered first. So they are actually named after their discoveries. So P700, the photosystem that contains P700, was actually discovered first, so that's why it is called photosystem 1, while P680, the photosystem that absorbs the light of 680, is photosystem 2 because it was discovered later. So it is not because of any other reasons, it is just because of their discoveries. Okay, so you don't have to get confused why is it not called photosystem 1 and why is it not called photosystem 2. The reason is because like this was discovered first, P700 was discovered first, that's why it is called PS1, photosystem 1. So first of all, let's start the fold reaction. So first of all, the light will fall on the photosystem 1 first. So what will happen when the light falls on photosystem 2, there would be two types of things that will happen. First, there would be a water molecule. The water molecule will break down and it will release oxygen molecule, half oxygen molecule, and hydrogen ions. And these hydrogen ions will be released and you will get two electrons that will be absorbed by the photosystem, and these electrons will move and they will be transported to the first primary electron acceptor. The primary electron acceptors will gain those electrons. After the electrons are given to the primary electron acceptors, the primary electron acceptor will move these electrons to a series of chains. As you can see here, there are different kind of blue bubbles in front of you, and we are going to fill them one by one. The first bubble is actually pheophytin. The first bubble is called as pheophytin. So this is the first electron acceptor. The electron will go to the pheophytin first. After that, the secondary acceptor is plastoquinone. It is plastoquinone. And the next electron acceptor is cytochrome complex. It is cytochrome complex. And the last electron acceptor is plastocyanine. Now let's discuss all of them one by one. The first is pheophytin. So pheophytin is not mentioned in many of the books, like it is neglected in many of the books, but in latest books and in, you can say, higher level books, pheophytin is mentioned. So it is always better to memorize these kind of terms. So in many books, the primary electron acceptor will transfer electrons to plastoquinone, then the electrons will go to cytochrome complex and then to plastocyanine. But pheophytin is also in between these molecules. So the first acceptor is pheophytin, and then the electrons will go to plastoquinone and then to cytochrome complex and then to plastocyanine. As you can see here, the electrons are moving from high energy. As you can see here, the energy is going downward towards high to low. So when electrons move from high to low energy, what will happen? Energy will be released, and the released energy will be saved in the form of ATP. So as you can see here, as electron is moving from high to low, the energy will be released, and the energy that is released will be saved in the form of ATP molecules. So the electrons will go from primary electron acceptor to photosystem 1, and from photosystem 1 it will go towards the secondary primary electron acceptor. So now the primary electron acceptor of photosystem 1 will gain those electrons again, and after primary electron acceptors of photosystem 1, it will go towards the ferredoxin. So now the electron acceptor is ferredoxin. After ferredoxin, the electrons will go to NAD positive. The NAD positive will receive those electrons and it will become NADH. with H hydrogen ions and electrons, the NAD positive will be converted into NADH. Now we have two types of products. First we made ATP and then we made NADH. So these are the two products that have been formed until now. So let's make the whole reaction again and understand the whole thing. So first of all, where did the electron started? The first electron was accepted by what was the first electron accepted? It was photosystem 2. After photosystem 2, the electrons went to the primary electron acceptors, right? After primary electron acceptor, the electrons went to the pheophytin and then it went to plastoquinone. I am skipping pheophytin because it is not mentioned in many books, but you still have to remember it, okay? And then it will go towards cytochrome complex, right? After cytochrome complex, the electron will move toward plastocyanin, and after plastocyanin it will go towards photosystem 1, and after photosystem 1, where will it will go? It will go towards primary electron acceptors, and after primary electron acceptors it will go towards where it will go? It will go towards ferredoxin, right? And after ferredoxin it will go towards NAD positive, and NADP positive will be converted into NADPH, right? So what is the difference between NADH and NADPH? So you have to be really careful when you are talking about respiration and photosynthesis. NADPH is the term that you will use in photosynthesis, while NADH is the term that you will use in respiration. So P is for NADPH that you can use in plants, and NADH is the term that you can use in respiration. So there is no such really great difference that you have to be really confused about what is difference between NADH and what is the difference between NADPH. So NADPH you can use the term in photosynthesis in plants, and NADH is the term that you can use in respiration and in animals. So this is the whole process that is right in front of you. So this is the whole cycle for non-cyclic photophosphorylation. So this was all about non-cyclic photophosphorylation. I hope you had understood until now what is happening here. So let's talk about some small steps that are in front of you. So how does photolysis of water actually occurs? So when sunlight strikes the photosystem 2, it excites the electron to a higher energy state. So the electrons will absorb those energy, and this energy will also be used to break down the water molecules. The water molecules, as in front of you, will be converted into hydrogen ions, oxygen atom and electrons. So in a single cycle there would be 2 electrons. As you can see here, there were 2 electrons that were being used until now, 2 electrons and then 2 electrons again. So these 2 electrons have been moving from photosystem 2 to photosystem 1. And what is actually happening at every single step? Most of the students get these MCQs wrong, so you have to be really careful about what I'm going to talk about. So first of all, as you can see, the first electrons moves from photosystem 2 to plastoquinone. So what happened here? You have to tell me first of all, photosystem 2 was oxidized, right? Photosystem 2 was oxidized because electron was removed, and plastoquinone accepted the electrons, so plastoquinone was reduced. After, plastoquinone gave electrons to cytochrome complex, so plastoquinone is now oxidized, right? And cytochrome complex is reduced. Then cytochrome complex gave those electrons to plastocyanin, so now the cytochrome complex is oxidized and plastocyanin is reduced. You can see the cycle here. So the atom or molecule or any compound that loses electron is oxidized. So all of these molecules here, as you can see, are getting oxidized and reduced at the same time, depending on the time of reaction. First of all, photosystem 2 is reduced, and then when electrons are given to primary electron receptor, then photosystem 2 is oxidized and primary electron receptor is reduced. Similarly, this whole chain will move until the electrons are accepted by NADH, and ultimately NADH is reduced. NADH is the final reduction product. So that's how the whole process will happen. So you have to be really careful about solving these kind of MCQs, like which compound is being reduced and which is being oxidized. So now you understand what is oxidized and what is being reduced. Let's move on to our next topic, that is about non-cyclic photophosphorylation and cyclic photophosphorylation. So in cyclic photophosphorylation, what actually happens, as you can see here, in non-cyclic photophosphorylation, that was actually just like Z scheme. So in non-cyclic photophosphorylation, it is also called as Z scheme, because as you can see here, it is just like a Z, right, in just like in alphabets. So in Z scheme, the final product was NADPH. As you can see here, the diagram has been like neglected and shortened, so you don't have to worry about, you just have to understand the concept. So the final product was actually NADPH, while after being following the Z scheme, the final product was NADPH. So after NADPH, the reaction actually stops, but there is a process that is actually cyclic photophosphorylation. In that process, the NADPH actually is not made. So what happens there is that when you need high amount of ATP, actually after the electrons are accepted by ferredoxin, the ferredoxin actually use those electrons and go back to the photosystem, and then it will start a cyclic chain reaction, and the reaction will keep on going on in a cycle, and ATP will keep on producing again and again and again. So ferredoxin is the point where the reaction will start the chain reaction. So instead of electron transfer from ferredoxin to NADP positive, the electrons will actually go back to the photosystem, and the chain will go on again and again and again, and ATP molecules will be forming continuously. So that is the difference between cyclic and non-cyclic photophosphorylation. So this is the major table that is in front of you. So in cyclic photophosphorylation, the process is actually cyclic, and in non-cyclic, obviously it is linear. So what are the photosystem involved? So in cyclic phosphorylation, only photosystem 1 is involved. We won't go back to photosystem 2. We only use photosystem 1, while in non-cyclic photophosphorylation you use photosystem 1, photosystem 2. In cyclic photophosphorylation, there would be no electron water splitting, because you won't be going back to photosystem 2, right? You only come back to photosystem 1, and the whole process will keep on repeating again and again and again. If you let me show the diagram here. So what actually happens here is that, for example, the ferredoxin receives the electrons, so the ferredoxin will use those electrons and it will go back and give those electrons to cytochrome complex. The cytochrome complex will give them to plastocyanin, and plastocyanin will give it to photosystem 1, and that's how the cycle will keep on going again and again. Ferredoxin will go back to cytochrome complex will give it to photosystem 1, and then that's how cyclic photophosphorylation will keep on going. So in as you can see, in cyclic photophosphorylation, photosystem 2 is not being used and water is not being broken down. So that's why, where does the electron actually came from? Well, the electrons actually came from the photosystem 2 that was actually in the non-cyclic photophosphorylation. So for cyclic photophosphorylation, non-cyclic photophosphorylation has to occur first. So if non-cyclic photophosphorylation is happening, and you have, for example, if you have low amount of ATP, normal process is non-cyclic photophosphorylation, and it is happening. And imagine you need high amount of ATP molecules. The plant needs high amount of ATP molecules. What will happen? Instead of making NADH, the process will shift towards cyclic photophosphorylation, and to make high amount of ATP molecules, the cyclic photophosphorylation will neglect NADH, and it will keep moving in a cycle again and again, and it will keep making ATP molecules. So that's how non-cyclic and cyclic photophosphorylation actually happens. I hope the cyclic and non-cyclic photophosphorylation concept is clear until now. So is oxygen produced? No, because obviously because water didn't split it, the water breakdown didn't happen, so oxygen will not be produced in cyclic photophosphorylation. What about NADPH? No, it will not be formed because the cycle will go back after ferredoxin. Will ATP will be produced? Yes. Cyclic photophosphorylation actually happened to produce high amount of ATP molecules, right? Because we wanted ATP, that's why cyclic photophosphorylation happened. And during cyclic phosphorylation, NADPH actually decreases. What about the primary function? It is primary function to generate high amount of ATP. What are the conditions? Well, conditions is high light intensity, water stress, and nutrient deficiency. Well, there is a deficiency of ATP, cyclic photophosphorylation will happen. And non-cyclic photophosphorylation is a normal process, while cyclic is a exceptional process. I hope I made it really clear. If you haven't taken the screenshot, take it, and then we will move to next difference between photosystem 1 and photosystem 2. So the difference is right in front of you, the difference between photosystem 2 and photosystem 1. So photosystem 2 is actually P680 that we discussed before. It absorbs the light of 680 nanometer, while photosystem 2 absorbs the light of 700 nanometer. Their position, they are right, we discussed them in your diagram, they are really clear. The electron source for photosystem 2 is water, right? The photosystem 2 gained electrons from the water. After water was broken down, the photosystem 2 accepted its electron, while photosystem 1 actually accepts the electrons from photosystem 2, and from photosystem 2 it goes through a chain reaction, and from plastocyanin it will goes towards the photosystem 1, right? And from about the products, you can say that the electrons are made by photosystem 2 and they will be moved towards the plastoquinone, and the final product of photosystem 1 is NADPH. Is oxygen produced? Yes. For photosystem 1, no, there is no oxygen produced. So about cyclic, the photosystem 2 is not involved in cyclic photophosphorylation, while photosystem 1 is involved in cyclic photophosphorylation. If we talk about associated pigments, then the chlorophyll A, chlorophyll B, and B carotenoids are involved in photosystem 2, and chlorophyll A and B and carotenoids are involved in photosystem 1. What about the summary of light dependent reaction? So in the light dependent reaction, ATP was made, and NADPH was made, oxygen was made, and hydrogen positive ions was made. So these are the products, and where are they made and what will they will be used for. So the products that are made in light dependent reaction will be used up in Calvin cycle, while oxygen will be released. So as you can see here, that's how oxygen is made. That's why plants releases oxygen. So the oxygen made during this process will be released in the air, and this oxygen is the oxygen that you are breathing right now. And the hydrogen ions that were made, they are used to make ATP molecules, because they were used in water splitting and drive ATP synthesis. Do you remember a chemiosmosis in respiration, right? You know that there was a gradient of hydrogen positive ions, and they moved from ATP synthetase or F1 particle from high to low, and ATP was formed, right? And that's how also happens in the light dependent reaction in plants. So the H positive proton gradient also helps in plants to make ATP. This process also happens in the plants. So that's the process that happens in plants and also in animals. So let's move on to our next topic, that is the light independent reaction. So light independent reaction actually happens in the stroma of the chloroplast. So this process is really important, and this phase actually requires NADPH, ATP, and carbon dioxide that were made in the light independent reactions. So it is divided into 3 stages. The first stage is carbon fixation, carbon reduction, and regeneration of RuBP molecule. So it actually contains 3 stages. The first is carbon fixation, the next is reduction, and then regeneration of ribulose bisphosphate. The names might seem really hard at first, but after you have practiced it again and again, you will slowly memorize these names. So first of all, the first thing that will happen is that the carbon dioxide that was made is being used here. So the carbon dioxide, the plants that need, they will take it from the air and it will be used in the Calvin cycle. So the carbon dioxide will combine together with a 5 carbon compound. The 5 carbon compound is ribulose bisphosphate. So the first step is that carbon dioxide will combine together with a 5 carbon compound, that is ribulose bisphosphate, the 6 molecules of ribulose bisphosphate that contains 5 carbon atoms. So 5 carbon ribulose bisphosphate, that is also called as RUBP, will combine together with a carbon dioxide. As you can see, the 5 carbon compound will combine together with carbon dioxide. Obviously it will form a 6 carbon compound, and then 6 carbon compound will be broken down into 2 molecules of 3 carbon compounds. First of all, 6 carbon compound is being made, and immediately this short lived intermediate compound will be broken down into 2 molecules of 3 carbon compound, that is 3-phosphoglycerate. This is also called as 3-PGA. So the 3-PGA will be converted into another 3 carbon compound, that is called as 3-BPG, 1,3-bisphosphoglycerate, and then the 3 carbon compound will be converted into another 3 carbon compound, that is called as glyceraldehyde 3-phosphate, and glyceraldehyde 3-phosphate will be given out, and remaining glyceraldehyde 3-phosphate will be used to combine together to form ribulose bisphosphate. So this was the general reaction. So now let's discuss all of these steps again one by one. So first of all, we discussed about ribulose bisphosphate. We will call it RUBP to make it easier to memorize. So RUBP contained 5 carbon atoms, and after carbon dioxide was combined with each, so 5 carbons and 1 carbon of carbon dioxide, it will form a 6 carbon compound. You don't have to memorize the name of 6 carbon compound. You can just jump towards the 2-3 carbon compounds, that is 3-PGA. So the 3-PGA has phosphate groups attached with it. As you can see here, 3-phosphoglycerate contains phosphate at carbon number 3. So it will be converted into 1,3-bisphosphoglycerate. So 1,3-bisphosphoglycerate contains 2 phosphate compounds. So where did the other phosphate actually came from? It actually came from the ATP molecules. So 12 ATP molecules were used. So 12 ATP molecules gave out 12 phosphate groups, and 12 phosphate groups were connected with the 3-phosphoglycerate molecule, and you got 1,3-bisphosphoglycerate, and the ATP was converted into ADP. And then the next reaction is 1,3-bisphosphoglycerate is converted into glyceraldehyde 3-phosphate, and this is actually a reduction process. Why is that? Because as you can see here, the NADPH actually gives out its hydrogen atoms. Hydrogen atoms are removed from NADPH, and NADPH is converted into NADP positive. As it is reduced, the hydrogens are given out to 1,3-bisphosphoglycerate, and 1,3-bisphosphoglycerate is converted into 3-glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate is actually a reduced process. That's why stage 2 is called reduction, and stage 1 is called what? It is called as carbon fixation, because carbon is fixed in RUBP. After G3P is being made, you will get out 2-3 carbon compounds. So what is the formula of glucose? Glucose contains 6 carbons, right? C6H12O6. So to make a glucose molecule, you actually need 2-3 carbon molecules. So you will take out 2 G3P molecules, and 2 G3P molecules will be used to make glucose and other types of sugars like glucose and other sugars. But major product is always glucose, and the remaining glyceraldehyde 3-phosphate molecules, that are 3 carbon molecules, will be converted into ribulose bisphosphate. And in this process, more 6 ATP molecules will be used. So 12 ATP molecules were used here, and 6 ATP molecules were used here, and 12 NADPH molecules were used here. So total how many ATPs and NADPH were used? 12 ATP plus 6 ATP. So you used 18 ATP molecules and you used 12 NADPH molecules in the whole process. So this was the whole process for Calvin cycle, and the first stage was carbon fixation, the carbon dioxide was added. The second stage was reduction, because NADPH was oxidized. NADPH was oxidized itself, but it reduced 1,3-bisphosphoglycerate. Okay, so NADPH itself was reduced. That's why it is called as oxidizing agent, reducing agent, right? So you have to be really careful about oxidation, reduction, oxidizing agent and reducing agent. So NADPH itself was oxidized and it reduced 1,3-bisphosphoglycerate. So 1,3-bisphosphoglycerate was reduced. So that's why NADPH was oxidized and it is the reducing agent, while 1,3-bisphosphoglycerate was reduced and it is the 1,3-bisphosphoglycerate is the oxidizing agent. So that was all about Calvin cycle. You can take a screenshot of the processes that happen in the 3 stages. Now let's move on to the stoichiometry of Calvin cycle. That is the most important thing, like how many carbon dioxide were used, like 6 carbon dioxide were used. How many RUBP was used? Yes, 3 RUBP was actually used and 6 RUBP was also produced at the end. So you can say the net RUBP is 0. What about 3-phosphoglycerate? Well, 12 3-phosphoglycerates were actually produced, and if we talk about glyceraldehyde 3-phosphate, there were 12 molecules that were produced. 2 glyceraldehyde 2G3B molecules were used to make glucose and 10 G3B molecules were used again to make RUBP. What about ATP? There were 12 ATP molecules that were used. That is the important MCQ here. And 12 NADPH molecules were used. And how many glucose molecules were produced? 1 glucose were produced. So to make 1 glucose molecule, you need 18 ATP, 12 NADPH and 6 carbon dioxide molecules. So that's how 1 glucose molecule is being made from Calvin cycle. So that was all about Calvin cycle. The last MCQs that you have to remember is that there is an enzyme that was used here, that is the Rubisco enzyme. So you have to know that Rubisco is like the most abundant protein that is present on planet Earth. Like if you want to know what is the most abundant protein, like it is 100%, no doubt about it, it is the Rubisco. Rubisco is the most abundant protein and most abundant enzyme on planet Earth, right? So it was one of like a really important MCQ. So if we talk about the factors affecting the rate of photosynthesis, the rate of photosynthesis is actually controlled by some of these limiting factors. The first is light intensity. You know that the greater the light, the more will be the Calvin cycle, right? If we talk about carbon dioxide concentration, it is also directly proportional. Higher carbon dioxide will also produce more photosynthesis. If we talk about temperature, the temperature, if you keep increasing temperature, the photosynthesis will also increase until it will reach our optimum temperature. What is the optimum temperature? It is 35 degrees Celsius. If we talk about water, then like water is not a really good limiting reaction, because like water affects the photosynthesis. Obviously it affects that, but it does not affect as much as light, carbon dioxide and temperature. So it actually affects photosynthesis but not as much as other factors. If we talk about wavelength of light, then yes, wavelength of light is also really important. So one thing that we left behind was action spectrum and absorption spectrum. So if you want to discuss about what is the effect of light, how light affects the rate of photosynthesis, then you can refer to your Cambridge or Pearson books, because this topic was really low yield, so I didn't discuss it in your lecture, but I highly recommend it that do give it a read from your books, from your Cambridge A levels book, or you can also go towards your Pearson books. So do read it out. Even if you want to read it from your national books, it's up to you. but do read out about action spectrum and absorption spectrum. So the wavelength of light that are most important are red spectrum and blue spectrum. So red and blue spectrum are the spectrums that are the most important one, and in these spectrums the photosynthesis is maximum. For all the other light spectrums the photosynthesis is minimum, but for red and blue it is maximum. So there are actually two types of plants. There is C3 and there is C4 plants. So C4 plants are like evolved versions of plants that are present right now. So C4 plants are the plants that reduce photorespiration. So C3 plants actually perform high amount of photorespiration, while C4 plants does not perform photorespiration. Photorespiration is obviously because it is bad for the plants. So if we talk about C3 plants, the final products for C3 plants is 3-phosphoglycerate, but for C4 plants they actually have the final product, the first product, as oxaloacetate or malate. It is a 4-carbon compound. The enzyme for fixation of C3 plants is rubisco. C4 plants also use rubisco, but they also use PEP carboxylase, that is also known as PEP. You have already discussed it in phosphoenol pyruvate carboxylase that was used in your glycolysis process, right? And the other difference is location of Calvin cycle. In C3 plants it happens in mesophyll cells, while in C4 plants it happens in the bundle of sheet cells. For photorespiration, yes, C3 plants actually follow photorespiration, while C4 plants does not do photorespiration. But even if they do photorespiration, the photorespiration is like really minimal and it is like close to zero. Okay, so that's why C4 plants are more efficient and they have higher survival rate. Well, because photorespiration is really bad for plant health, and photorespiration actually negatively affects plants. Discuss about photorespiration, do refer out what is photorespiration in your books. I haven't discussed low yield topics in this lecture because I thought that this chapter is already very big, and if we had to discuss every single point, then this lecture would have been like 2 to 3 hours long. So we just stick to the high yield topics. The examples of C3 plants are wheat, rice and spinach, and for C4 plants obviously maize and sugarcane. These are examples for C4 plants. C3 and C4 plants is also low yield, but we still discussed it. Let's talk about photosynthetic pigments. We have already discussed about chlorophyll A and chlorophyll B. So chlorophyll A is actually blue green and chlorophyll B is yellow green. So what does it mean? So blue green and yellow green actually means they reflect these pigments. They don't absorb these pigments, but they reflect these light. As you know that what is the spectrum of light? It is actually VIBGYOR, right? What is VIBGYOR? VIBGYOR means violet, indigo, blue, green, yellow, orange and red. So VIBGYOR is the question that you have to remember in your IMAT exam. So this is the spectrum of light that is being used in light. So violet, indigo, blue, green, yellow, orange and red. So in this spectrum, blue and green is the light that is reflected, while violet, indigo, yellow, orange and red is absorbed by it. While for chlorophyll B, yellow and green is reflected and the other all light spectrum will be absorbed. So what about green color of plants? The green color of plants is actually because of chlorophyll molecules. Because for example, if it is a leaf in front of you, right? If this is a leaf and light falls on it, and if there is a spectrum of VIBGYOR in the sunlight, then all of the light spectrums will be absorbed, but the spectrum that is not absorbed is actually the green spectrum, for example, and green color is reflected, and the reflected spectrum will be the color that you will see, right? So all the other color will be absorbed and the green spectrum will be seen. This is an example, right? So the color that you see in chlorophyll A is actually blue and green. Why is that? Because the all other spectrums are actually absorbed. So the wavelengths that are absorbed are blue and red, the 430 nanometer and 662 nanometer. And for chlorophyll B, the blue and red, the same wavelength are absorbed. I think something is like wrong with my laptop today, so you have to please like sometimes it can happen. Okay, you have to be like a bit patient. If this error comes again and again, I will work on it. I don't know why is it happening. Like I will get it repaired today, so bear with me. Okay, so the other pigments are B carotenes, xanthophylls, and the last is phycoerythrin. So these are the important pigments. The most important are the first four, the carotenes and chlorophyll A and chlorophyll B. So these are the important pigments that you have to keep in mind. The table is right in front of you, so keep in mind like the action spectrum, the absorption spectrum, study about these pigments. Though these are not that high yield, the high yield topics we have already discussed. So this is the comparison table between respiration and photosynthesis. So the photosynthesis and respiration, this is a really high yield topic, because now you know that in photosynthesis you actually make glucose and carbon dioxide and water is used, while in respiration it is actually the opposite that is happening. So both of these reactions are really important. Photosynthesis wants carbon dioxide and water while it makes glucose, and respiration wants glucose and it makes carbon dioxide and water. So both are dependent on each other. So this is the table right in front of you. Take a screenshot of it, and then you can see the diagram here. The cellular respiration is producing what? It is producing carbon dioxide and water that is being used in photosynthesis, and photosynthesis make glucose that is being used by mitochondria in respiration. And this is the whole process that is occurring in a cycle. The last table that is in front of you is the difference between light dependent and light independent reaction. We have already discussed. Let's discuss it for the last time. I swear this is the last time, because this is a high yield topic. It is really important. So light dependent reaction happens on your thylakoid membrane, while a light independent reaction happens on stroma. I am going to say it again. Light independent reaction means it happens in without light. It does not mean that it does not happen at all. Light dependent reaction means it happens in the presence of light. It does not mean that it only happens during light, while light independent reaction can happen without light. It does not mean that it does not happen in the day, because some students are like, dark reaction only happens during the dark. No, dark reaction also happens in the daytime, but the thing is that they can also happen without light, right? They are independent of light. So next thing is that, does light dependent reactions require light? Yes. Light independent? No. Inputs, we have already discussed that the inputs as in front of you is that light dependent reactions requires NADP positive and ADP. They produce ATP. So the products of dark reaction will be used in light reaction, and the products of light reaction will be used in dark reaction. So this cycle will be continuous and keep on going again and again. If we talk about inputs of light reaction, as you can see, the input is water and light, and NADP positive and ADP positive. And if we talk about the inputs of dark reaction, it is carbon dioxide and ATP and NADPH, right? And outputs are right in front of you. The output of light reaction is oxygen that will be released in the air, and ATP and NADPH that will be used in dark cycle. And what is the output of Calvin cycle? The output of Calvin cycle is glucose, sugar, and NADP positive and ADP positive that will be used in light reaction. So as you can see here, all of it is connected. So that was all about light reaction and dark reaction. Before you go, listen to my last one advice, that is, this chapter is not really occurring after 2023. Like MUR is not giving focus to this chapter. but don't skip this chapter. Like you have already taken the lecture, you have already discussed about light dependent and light independent reaction. Why not just work on this chapter a bit more? Like I am not telling you to go all out on this chapter, but still don't neglect it. This chapter is still very important. In Cambridge era before 2023, this chapter was really important, but after the paper pattern has changed, this chapter is still really important. Okay, this chapter might come in 2026, in 2027, in 2028, in 2029, in 2030. This chapter will still remain an important chapter, but don't neglect it. But still, most of your energy should go towards the respiration side, but still don't leave this chapter, don't leave these topics. We have already discussed about what are the high yield topics and what are the low yield topics. They are in front of you. Your main focus should be on high yield topics. For example, if you are in your last weeks or last month of your preparation, don't go and revise every single thing. Don't go towards C3 and C4 plants. Don't go towards action and absorption spectrum. Don't go towards like studying about pigments and all of that stuff. You should be focusing on the light dependent and light independent reaction, about what is chloroplast, about chlorophyll. These are the high yield topics. Go towards these topics and not towards the high yield topics in your last days. In your last days, this chapter focus should be on the high yield topics. While for the other chapters, obviously you should be revising the whole respiration chapter, the whole cell structure function chapter, and whole biological molecule chapter should be revised in your last days, because those are the most high yield topics. You shouldn't be skipping anything in your physiology part, right? So the physiology part is going to start really soon. In the next lecture we are going to be starting your DNA and your central dogma section, and it will be like a bit long sessions, about maybe it will be divided into 3 to 4 chapters, and then we will switch towards the last section, that is physiology and anatomy section. So this was all from my side. If you have still any question, you can ask them in the the comment section in the basement. If you are on YouTube or if you are on the paid session, you can just write them down and I will answer all of your questions. Okay, so that was all from my side. Take care. Bye bye. Allah Hafiz.